First of all, thank you for having me here. Can you guys hear me all right?
Yeah.
Yeah? All right. So today I'm going to talk about basically using a lot of public data and how to make sense of it and how to use it together in tandem. I like to call it free search. It's a good starting point for labs or PIs that don't have a lot of money and you can't do your own experiments. And it's also good if you have a lot of money and you want to know what to look at. So let's see. As has already been touched upon, there are just tons of data that's becoming public. If you receive NIH funding or a lot of other federal funding now, you have to make your data available, though some people try not to. So anyway, that's another story. So what we have here plotted, and it just sort of reiterates what we've been showing, is just a number of data sets in black that have at least 10 samples and have the word control in them. So very rudimentary filter from what's available from GEO and NCBI. And you can see we have plenty of those, and these are the corresponding number of samples that go with these. So there's over 300,000 samples. And so what we want to do is make use of this data to, well, basically for our own purposes. We want to focus on blood profiling in immunologically relevant data sets. We want to do biomarker discovery that will help us distinguish patients, different subtypes of patients, understand diseases, and even develop some clinical assays. And so that all sounds great, but there's a lot of huge caveats, right? So first off, we're looking at different platforms. And so some of these platforms have 44,000 probes, some have 22,000 probes, and actually, I want to go back one slide, some of these platforms are now RNA-seq. So that's our guess about what's happening here. This is all from GEO, and so people we think are starting to switch over from microarray to RNA-seq right around here, and you can see just from the number of poster presentations that are referencing RNA-seq that that's starting to happen, which is awesome. So you have this issue where you have different platforms, you have different technologies, if you have different probe numbers, different number of probes being mapped to different genes. Some genes may be missing probes, and then you even have different technologies. So if you look at Agilent, you have two-channel arrays, and you have single-channel arrays. If you look at Illumina and Affy, although Agilent is now jumping on board, and their newest chip you can use as a single-channel microarray. So you have to be careful if you ever use any of their public data, make sure which way it was used. And then, of course, even with the same platforms, you have to worry about batch effects. So what we're going to show are module-based methods that can help mitigate these and let you incorporate different data sets from different platforms. And what's a module? Basically, as was referred to before in Hulin's talk, it's just a bunch of genes that move up and down together. I'm going to refer to a lot of them here. I'm not going to talk, unfortunately, a lot about how they were built. A lot of that work was done at Baylor University, not at Benaroya, by Nicole Baldwin. And our idea here is to basically use modules which are made up of groups of genes or groups of probes to have a robust measurement. So that means that if you have one platform and it has 100 probes in a module and the next platform only has 75, the overall movement of that module won't be too affected. And some nice side effects of this, usually side effects aren't nice, you get to view the data a lot more concisely. So instead of looking at tens of thousands of probes, we look at hundreds of modules, which is much easier to look at. And then you can easily have some biological information associated with those modules. And we don't do much modeling. I'm not going to talk about it too much. But if you want to use these as input, as parameters, you then start to address this cursor dimensionality. If you have hundreds of samples and hundreds of modules to look at, you can start to think about modeling. So these modules, when I'm not going to tell you how they were made, they are basically data-driven. So we all know about gene sets from gene ontology that you could look at. You could also take that approach here, but that's knowledge-driven. We're aiming at data-driven. So the idea is that we want to perturb the immune system, right? So Hulin was talking about, I think he said disturbing the steady state. So it's the same idea, perturbing the immune system. And specifically, we want to do it in some categories here. Sorry. And so we want to do it with infections. So in that case, we have TB, HIV, and some sepsis data sets. We want to perturb the autoimmune sense, so we have lupus, 2 lupus studies, and we have systemic onset juvenile idiopathic arthritis. Then we also want to look at immune deficiency, so we have a B-cell study. You could also think of HIV as immune deficiency. And then lastly, we have tolerance or immune transplant. And so what happens there is we have 410 samples, but what's important is these were all run at the same facility at the same time. They all have matched controls, right? So it's going to mitigate batch effects you would expect to see. And so we can sort of look at what's sort of a theoretical map of how the immune system moves up and down in large scale. This is by no means every possible perturbation, right? So that then, we do that all in Illumina using the same exact chip type, platform type. And so the idea is we want to then map anything we can to it that maps reasonably well. So we just map by RevSeq. It's not particularly sophisticated. If you use Bioconductor, there's a lot of good databases per platform, and they allow you to map pretty easily. And so doing that, we're able to not just look at the V2 Illumina that was used to generate the modules in the first place, we're also able to look at V3 and V4, and then also a lot of Affymetrix data, which is good because it's one of the most popular. So HGU 133A and on. So that's great. And then we also can do the newest adjuvant maps all right, and the ABI V2 maps okay. If it doesn't map particularly well, we don't support it. And we are also working on right now, as a lot of people are, is RNA-seq, and that's pretty easy to map because it covers so much, right? So when you do this, essentially you get this surrogate array. You're all mapping to the same sort of coordinates, right? And we end up with this 260 modules, and they basically allow us to summarize any of these platforms. But to do this, if you're going to look at a single study and compare it to the modules you've already made, you need to have, make sure you have good controls and basically a very simplistic design. We don't do longitudinal data, which is a big drawback of this. Obvious question early on is how well do these different probes really move up and down together, right? So an earlier version of these modules was made actually starting with Affy back in 2008. And so this is an example if you look at healthy and people who received liver transplant. And so in one module it's defined, and after the fact we go and annotate them so you could have some biological meaning. These are the probes and their movement across healthy and transplant samples. So it's a little chaotic. And all the samples were done using the HGU 133A array. And then the same exact samples were done using the Illumina. And you can see it's pretty old because it's back when the Illumina arrays were called Centrix. And so if you sum it up and you take for each sample the average of the probes in that sample for this module, you can see that they move up and down together pretty well. And so you have the blue here in Affymetrix, and the orange, I'm colorblind, but I think that's orange. Here is the Illumina. And so it's a pretty good correlation, showing that you do have good mapping and good consistency between platform types. And that's not just a one-off. I know a lot of times people can wonder if you're just picking your best module. That's actually not the best module. This is an even better module here. Same sort of plots, and you can see we have a correlation of 0.98. So this is generally what you see between different platforms. So what I want to do now is go ahead and show you. Right now you're probably still wondering what this looks like. I'm going to show an example that will help clarify what's actually being done here. So this is a use case where we want to help clinicians. So when a clinician sees a patient and they have a respiratory, especially an acute respiratory infection, they don't know if it's caused by a bacteria or if it's caused by a virus. And so if they knew that, that would quickly determine how they would treat the patient. If you had a patient with a bacterial infection, you want to give them antibodies pretty quickly. If you think somebody has a viral infection, but they actually have a bacterial infection, there can be pretty negative consequences when you don't give them the antibodies that they need. And then on top of that, if you know somebody has a viral infection, or at least you think they do, you want to know if they have a specific type, which is called RSV. It's particularly acute, and you would want to watch them better, especially in children, because you could have pretty negative consequences again. and you probably want to keep them hospitalized longer. So clinicians would really love to be able to tell the difference. So what we want to do is use this module-based approach in order to bring in as many data sets as we can and eventually identify modules that are able to separate people who have an infection that's due to bacteria or they have a respiratory illness due to virus. And so then we can go develop like maybe a qPCR assay using genes that are inside of those modules. So here, what you can notice already is that we have multiple chip types. Not only do we have Illumina and AFI, but we have different versions of AFI. And then we also have, some studies some studies just have bacterial and some have viral and some have both. And then you can see our cell types. Some of it's whole blood and some of it's PBMC. The modules were all made using whole blood. So this is actually already been reduced for viewing purposes, and you can see it's not that great to look at. But what you have here is each column is a sample, right? And across this way are modules. And so instead of showing you 260, we're just showing you about 22 of the most variable. And the red color is indicative of the probes that are in that module, what % are up. And the blue is what % are down. So if you have an Illumina microarray and this particular module has 100 probes in it, then that's telling you how many of those probes are up. And if you have an AFI and it only has, again, 75, it tells you what % are up or down. So if something's down, it means it's actually higher expressed in the controls for that particular experiment or that particular study. And if you zoom in, these colors here indicate which study the sample came from. So you can see that they interlace nicely. You don't see that one study is all off on its own and another study is separate. So that's a very good trait. And then if we just go ahead and look at which samples are bacterial, you can see they cluster nicely. You do have this out here, which is a little bit worrisome, but you still have a nice big chunk here. And then in the middle, with a pretty variable expression in two separate branches, you have all the virals. So you can see that already you can break out, and they're not breaking out by study. And for display purposes and keeping things simple, these don't break out by age, they don't break out by gender. We check these things out. These break out by disease. So, actually, let's go back. This is pretty ugly. This only includes bacterial and viral. I did not include controls, and I didn't include the RSV just because it won't fit on the screen. This particular meta-analysis has over 300 samples. I've done others that start to encroach on 1,000, and it's even more impossible to look at. So what do we do to cut these branches and summarize them and look at them, right? As pointed out earlier, it's very difficult to decide how many clusters you want, but we have prior knowledge, and that is we have a labeling of our samples, whether or not they're control, whether they're viral, whether they're bacterial. So we use a really simplistic measure called the RAND index. And some of you are probably familiar with it. It's super easy, and all it does is look at every pairwise combination of samples. And so it's asking how often they're clustered properly together. So if you look at one sample and it's viral, and you look at another sample and it also is viral, you'd hope they end up in the same cluster, right? If you look at another sample and it's viral or another pair, and one is viral and one is bacterial or one is control, you'd hope they were in separate clusters. And that's all you're counting here with A and B. These are just the counts of when you have a pair and they have the same disease and they show up in the same cluster. And this is your count of when you have two different samples, one's in disease, one's a control, or different disease, and they're in different clusters, which is what you hope happens. And then you just divide by all your possible combinations and choose 2. So it's a simple measurement. It goes from 0 to 1, 1 being ideal, which of course you never see, and 0 being just basically chaotic and random. And so what we also do is to check that you don't have other factors that are dominating. You could do this by gender or by or other, or the study or things like that, and see if they have higher RAND indexes. That's a huge red flag. So when we do that to the study we just looked at, it goes down. There is a little connector up here. It goes into just 2 branches. What we do is we summarize these branches just using the first principle component, and we scale it. And we label up here the percent of the cluster that's made up of bacterial samples, RSV, and viral. So down here are the numbers, and you can see that this right-hand cluster is almost completely bacterial. All right? It's 82% bacterial. And then the left-hand cluster is made up of RSV and viral. RSV is a virus, so that's not surprising. And so that's 95% viral. So this is good for illustrating and visualization purposes, but it's not, you know, what I would call true prediction, though some people use this as for classification. You wouldn't want to develop an assay off this. Our RAND index is 0.25, which is pretty good. And we start to see certain modules that are interesting. We see that the viral samples tend to have an interferon response, which is interesting. And then we could even see that the inflammation is slightly higher in people with bacterial infections. So, like I said, that's not proper prediction. So what we use instead is we use random forests. And so I'm sure a lot of people here are familiar with random forests, but I wanted to just do a really simple intro to them. The name kind of says it all. So it's basically a classifier. It's a voting system, and it's built of many simplistic tree voters. So you have a lot of classifiers that are each contributing a vote, and then those many trees make your forest, and each of those trees is built using a random subset of your samples and a random subset of your predictors, or in this case, your modules. So that's why it's random. So you take a random subset of your samples and a random subset of your modules, and then you make a really simplistic tree. They can be more complex than this, of course, and you find out which module is the most useful. Then you rinse and repeat, and you do this over and over, and you get many, many simplistic classifiers that then vote, right? So what I really like about this is when you're doing collaborations and you're working with biologists that might not have a particularly strong mathematical background, it's a classifier that's pretty easy to explain to people, even if they don't know all the ins and outs. And one of the best parts of it is that you can intuitively understand why you're using certain modules and which ones are most important. They're simply the modules that are kind of used most often. There's a little more sophisticated than that, but it's which modules are used most often in all these trees. So the other really big advantage over something like an SVM or other predictors is that you have a much fewer modules that are selected as important. So if you can also do this using just probes on a single data set, and you still get many fewer probes selected than if you use other types of classifiers. So that said, if we go ahead and we look at the study we were just looking at, you can see that if we do our prediction, we get an error rate of about 15%. Most of that error is occurring in confusing RSV and viral samples, which is not surprising. Like before, we noticed that they're both virus, that RSV is a virus. When you go ahead and cluster or actually categorize RSV as just another virus, your error rate drops to about 11%, which is not perfect. And maybe you don't want to just jump off and make your assay just from this. But what we're going to do now is work with clinicians to develop a qPCR assay using a fluid eye machine. And we're going to select, this is just using 20 modules. So we'll probably take 4 probes, 4 more central probes from each of those modules, and use those in a qPCR assay on actual samples and see what kind of prediction we can get. First, we're going to start with viral versus bacterial, and then later on we'll look at RSV versus viral. So another example here is in the case of tuberculosis. So in tuberculosis, people who get it go through an acute phase, and then they transition to a latent phase. And a small subset of folks will reactivate. And so if somebody reactivates, you want to know for quarantine reasons and also for how you're going to treat this person. And you'd like to be able to tell the difference between somebody who was reactivating versus somebody who just happened to have a particularly bad cold or flu. So what we would like to do here is look at, make a meta-analysis and look at 6 data sets of folks with TB and controls. And in this case, we're looking at Agilent and Illumina. Our Illumina happen to all have the same exact chip type. And this is all whole blood, which is great. And so I kind of jumped the gun here a little bit, One study has no controls. So earlier I said that a study to be incorporated should have controls. Through previous analysis, we show that essentially controls and latent can't be separated. They look identical. And that was known. Clinicians weren't surprised at all. So to include one more data set, I went ahead and brought this data set in with its controls being latent, which is cheating. So we have over 300, actually we have exactly 300 samples. It just happened to be that way. And I just want to point out one thing. You can always tell when a bioinformatician's giving a talk because they present the geo number instead of the actual PI's last name. And so jumping ahead of those other analyses, like what I call a fingerprint plot with all 300 samples shown, I just go ahead and show the RAND index, and you can see it's not too hot, right? And that's because we have 3 labels. We have control, latent, and active. And so you can see that we're able to break out 84 samples that are active pretty well here. But then this one cluster, which is huge, it's over a third of our samples, 112 samples out of 300, is made up of 1/3, 1/3, and 1/3 each of the categories. It's not, a warm, fuzzy feeling. So what we do is if you label the controls and latents together, because clinically we know that they're essentially identical, although you could argue it's cheating, you see that they separate very nicely. And our RAND index jumps to 0.38, right? So we then are also able to look and see, hey, look, that interferon shows up again as being a high predictor between active TB and latent or control, which is interesting. When you do this analysis, it's not like interferon always shows up. It just happened with the last meta-analysis and this one, and that's the case. And then you can see the erythrocyte signature that's higher in controls and latents. So when you go ahead and you look at this with the random forest, you can see again that you don't do too hot when you have the 3 labels. You have high error rates for your latent and your control at 40%. That's never a good sign. But when you break it out and you call these control and latent, you get pretty good accuracy. So that really suggests that there really is a huge difficulty splitting up controls and latents. And so the clinicians aren't crazy. You probably have to do much more targeted studies if you want to go ahead and see what's actually happening there. So for future work, we want to develop these assays. We also want to incorporate RNA-seq into this. And a really strong critique of this is that when you have a disease, it affects your cell counts of certain subtypes. And that for some studies, you're not actually capturing you're actually capturing a change in the number of cells, maybe more or less erythrocytes. And so what we have some folks doing right now is some pretty time-consuming assays where they sort fresh blood into 7 different cell types. And that takes about 12 hours. It's pretty intensive. But long-term, we're hoping to do RNA-seq on those. And so that you could see that a specific, CD4 positive T cells, this is what their signature looks like, and they will have their own set of modules. So, there's a lot of talk about interdisciplinary teams. We have basically 3 groups. We have our bioinformaticians, our bio-IT or software people, and then we have our genomics core who's running all the microarrays and the RNA-seq, and then we have the always undercredited postdocs in the middle. And so I have to give a lot of credit to Dave Anderson, who helped me support a lot of these platforms. I always told him it was the last one and turned around and said, no, we're going to support this other platform. So he was very patient with me. And then Laurent and Noemi are clinicians, and they really helped drive why we were looking at certain studies that would be useful for clinicians. And lastly, Brad helped with a lot of the software. And I mentioned before that these original modules were designed by Nicole Baldwin at Baylor, but a lot of this is basically the starting point is the output from something we call MAT, or Module Analysis Tool, which will be released to the public in the following months. And a lot of that work was done by Elizabeth. A lot of it's based in R, and then our software team developed a web-based package around it. And so similarly, once that's released, we're going to be developing something that is tentatively titled MetaCat for Meta and Comparative Analysis Tool, which will employ these methods. Thanks.
Classification accuracy when you use modules versus individual genes?
No, we haven't. So I would imagine you could probably get better accuracy from individual genes from a specific data set with specific probes, with specific platform. So what we're more interested in is looking at, this was all about classification here, but another thing is to see if you could find new and novel groups of genes that are moving up and down because you're able to have a larger metadata set. So, but if I was to study classification specifically, ideally you'd get huge number of expression data and start from there with one platform and go from single genes.
I was wondering how much effect some of the clinical uncertainty might have on the results, because those co-infections might give fairly confusing signals. And I think those do happen in surprising frequency.
Yeah.
If you actually achieve 90% accuracy, you may actually be as good as you want to get.
That's
How do you way of getting at that?
That's very true. And so I had to rely on Noémie and Laurent a lot for that. But I got an email this morning telling me that there were many more than 3 flu infections here. But those were in this study. But those were the only 3 that had respiratory infections, that had an acute respiratory phenotype and didn't have co-infections. So you bring up a good point, and that's why we hope to develop the qPCR assay and look at a very specific population where we know exactly what happened with each of those patients.
But I'm wondering how you dealt with a problem that is a many-to-many mapping. So many probes to one red set or couple of chapters that we tend to use, and often there are many transfers mapping the same probe.
So that's a really hard issue, right? This mapping, and we have an argument over whether or not even to use RefSeq or Ensemble ID, and then you could really, if you wanted to, just start using probe sequences and where they're mapping specifically. So it's been a while, let's see. If we have a case where a probe, so you can have a case where you have a probe and a module for a specific RefSeq ID from the original Illumina V2. And if you bring in a new platform and the RefSeq for it, if it has, say, 3 or 4 probes, but they map to multiple modules, we just kick it out. We don't even use it. So it's a little overly strict. But about 70% of the time, 75% of the time, it works out so that all the probes from this particular RefSeq map into the same module, which is what you would hope, right? Because they're supposedly measuring the same thing.
And then you get that right.
No, when that happens, we just say that, we have a few cases where we might have had 100 probes in a module to start with, and we might have 105 from the different platform. But because everything's percent-based, it works out.
So correctly, you're quantifying the modules by the percentage either upregulated or down. So how do you, that can be platform dependent, right? How you define your cutoff. So then again, you have the problem of comparing between platforms. How do you?
So we need to look into that more. It looks like, I don't know how this happened. It looked like from Affy to Illumina, using the same exact cutoffs works fine. But the real question is Agilent. So for whatever reason, my Agilent data, not my data, but whenever I bring in Agilent data, it always looks brighter, whether it's more, like a higher percent that are down or higher percent that are up, which we need to return to and double check.
And related to that, when you were differentiating the bacterial and viral, it looked like a lot of the differences were quantitative rather than qualitative. And is that different? Is that separate by platform as well?
Can you be more specific about qualitative and quantitative in that case?
That a lot of the modules were moving in the same direction, and it was just stronger in bacterial or viral, slightly weaker, but same directionality.
Yes.
That can be platform differences?
Well, that's probably has to do with how the modules are made to begin with. So you get, they do rounds of selection, so they find modules that moved up and down across all 9 data sets. And then after they do that, they find modules that move together all across all 8 data sets. But you could easily see cases where, and you see that we have three modules called interferon, right? And those could easily be argued that they should be one module. I don't know if I'm answering your question there.
Okay, well, thank you very much, Mike.